Wonderful. Single cell analysis for individualizing therapy. Something I can't do that.
Okay. Good afternoon, everybody. First, I would like to thank the organizers for giving us the opportunity to present our work. So, as you can see, the title of my talk is Combining Machine Learning with Single Cell Analysis for Individualized Precision Medicine. I hope that at the end of this talk, I can convince you that it is now a reality, not just a dream. Cancer cells, they interact dynamically with their tumor microenvironment during tumor progression, thereby changing their phenotypic states. And this challenges our ability to properly or precisely diagnose and treat a given patient. Today, I'm going to present results from two studies. The first study is going to be about a computational framework called StateMap, which is abbreviated here as STAMP, which can be used to build a reference map to model plasticity of biological processes. And I'm going to focus on a biological process called EMT, which is a well-studied process that tumor cells exploit for invasion and metastasis. And I'm going to demonstrate how we can build a map using time-course single-cell analysis, and then use the project clinical samples on the map for clinical use. The second study is going to be about a published algorithm called DrugNem, which has a potential to optimize combination therapy for a given patient by accounting for early intracellular responses. And I'm going to demonstrate an example in leukemia. So why do we need a reference map? So a reference disease map could be an indicator for effective diagnosis and treatment. So for example, with respect to EMT, which is a well-studied phenomenon in biology, which is involved in developmental processes, if we can identify the beginning states of the disease, the intermediate states of the disease, as well as the end state of the disease in the form of a map, then we can then collect single-cell data from a given patient using a technology like CyTOF, which is a technology that can measure several proteins at a single-cell level. And then with such a data, if we can project it, depending on where the cells fall on the map, can give us an indication of how sensitive drugs are to the cells, assuming that we know their mutational profile. So to generate the map, we collected time-course data over a period of 20 days. So we stimulated cell lines using TGF-beta, which is a strong stimulant of EMT, and for over a period of 10 days. And after which we then withdraw TGF-beta for an additional period of 10 days. And so we have a span of 20 days where the continuous treatment and withdrawal of TGF-beta allows us to model the entire EMT and MET process. So here we have the panel of markers that were used and measured using CyTOF, and with this data we can then generate a map. The next slide summarizes the framework for STAMP. So basically, we pull the data, and then we partition the data using an algorithm which I developed during my postdoc at Stanford called CCAS, which stands for Clustering, Classification, and Sorting Tree. And then we partition, then we combine it with the Voronoi mapping, which allows us to create boundaries on the map. And at the same time, of course, using the famous neural network, which has been very powerful, we also train the neural network on a t-SNE feature space using the high-dimensional data, and this allows us to project new samples on the map. So ultimately, our goal is to project clinical samples on the map. So here we have 5 patient samples of non-small cell lung cancer. And if you look at the map for patient 1, we see the partitions where they are labeled. So we have, based on after projecting the time course data, we have 3 epithelial state markers, regions. We have denoted as E1, E2, E3. We have partial EMT state regions denoted as P, EMT 1, 2, and 3. And then we have a mesenchymal state denoted as M, and then we have a partial MET state, which is a reversible region denoted as MET. So if you look closely at the data, you will see that patient 3, 4, and 5 most of the data is represented in the epithelial and the partial states. And it seems that most of these patients are also correlated with poor differentiation status. And you could also see that based on their mutation status, there is some kind of a trend in their ordering. A second application of the map is to study drug response. So here, this is preliminary work. So here, this is a projection of the data, single-cell data, by treating the cells with elatinib, which is a drug that is used to treat lung cancer patients. And you could see that over a period of days, most of the cells stay within the epithelial state and the partial EMT state. So this really gives you the power of identifying certain resistant states, which can allow us to optimize combination therapy. However, currently, optimizing combination therapy does not account for intratumor heterogeneity at the single-cell level. So an optimal combination therapy should be able to control for intraspatial heterogeneity. It should be able to control for interspatial heterogeneity, since tumor cells can migrate from a primary site to a metastatic site. And it should also be able to control for temporal changes. So this motivates the need for an algorithm such as DrugNem, which is optimal for optimizing combination therapies for a given patient by optimizing early response in time after treatment. So here is just a framework for applying the DrugNem within a clinical setting. So if we were to collect, let's say, tumor samples from a patient, blood or solid tumors, then we dissociate the cells and we treat them with drugs denoted here as green, red, and blue, using a single-cell technologies like CyTOF. Then an optimal data for DrugNem should have markers that can identify lineages, intracellular markers, as well as a strong drug response marker, desired response. And with this, the algorithm is going to combine a subpopulation identification state, followed by an optimizing a drug network using the drug effects, and then it's going to use the network to score and rank the drug combinations. And with this ranking, it can then identify optimal combinations for the patient. So the slide summarizes you the underlying idea behind the DrugNem scoring or optimizing algorithm. So if we were to treat a sample with two drugs, green and red, then it is likely that some of the green effects will be nested within the red effects. Some of the red effects will be nested within the green effects. There could be an intersection of effects, or we could have a disjoint set of effects, or one of the drugs could show a strong marginal effect. So the idea of DrugNem is that it optimizes subset effects at the single-cell level. The objective of DrugNem is to identify drug combinations that offer the maximal response with the minimum number of drugs. So in this scenario where we treat the tumor with 3 drugs denoted here as green, red, and blue, and if we observe that the blue effects are a subset of the other 2 drug effects, such a scenario is not optimal. All we need is just the 2 drugs, green and red, which is going to target all the pathways and all the cells. So we then applied DrugNem to a cohort of 30 ALL patient data. So here we show an example of the result for one patient. So we collect blood from the patient, we treat it with 3 drugs, tofacitinib, dasatinib, and BZ234, and we then measure collect CyTOF data for these patients, and then use the lineage markers to identify 7 subpopulations for this particular patient. And then we use the intracellular markers to generate a drug network, which indicates here that the tofacitinib effects are a subset of the BEZ and the dasatinib effects, implying that the BEZ and the dasatinib combination is optimal for this patient. So we can do that for each patient. And so we have a distribution of response from DrugNem predictions. And clearly, 18 of the patients show DrugNem predicts that BEZ and dasatinib is optimal for 18. But if you look at the tail, we have these 2 patients that DrugNem says you can use only tofacitinib. So this is really showing that we can actually do individual patient prediction. So to conclude this presentation, I introduced 2 computational frameworks, STAM, which can be used to build a reference map to model the plasticity of biological processes. I then showed you an application in EMT, which can be used for clinical use for diagnosis and treatment and studying drug response. And I then also introduced you a second algorithm which is published called DrugName, which optimizes combination therapy for an individual patient by using early response data. And so, and I showed you how we can predict, use DrugName to predict optimized combination therapies for ALL patients. So you can see, if you want to talk more, there's a poster after this evening. And I would like to acknowledge all those, my collaborators who helped in this project, and special thanks goes to NIH and Chinese Zuckerberg Initiative who sponsored most of the work. Thank you, and I'll open for some few questions.
Thank you so much. Great talk. So I'm sure everyone's gasping for coffee, but again, time for a couple of burning questions.
Hello, just a quick question. So from these predictions, were these predictions validated in any way? Or how do you know that these predictions are correct?
Okay.
That's the first one. And then the second one is, you say you use a t-SNE projection to feed it into your neural network. What is the point of using already an embedding when you're doing, like the neural network is creating also an embedding, right?
Yes.
So why wouldn't just you feed the raw data instead of using the projection of
True.
t-SNE into
You
that?
mean the intermediate step?
Yeah.
Okay. So the first answer, this is one of the ways to directly, if you read the paper. So one of the ways to predict is that you go ahead, you do another site-off with the single drugs and combinations. And then you can then try to do the predictions from the algorithm in repeated rounds and try to see how good the predictions are by comparing with the real data from the two drug combinations. So here, for example, you see all the data with single drugs as well as combinations. So you could potentially do the predictions and then validate them by looking at how, for example, for this particular patient, we did like 90 rounds of DrugName, and using a mismatch analysis, you can potentially see how the algorithm behaves. Of course, that is one way. Another way is to actually also look at, do the single and see what happens over time. So this is more in vitro validation. That was more like using early response, but this is really using cell line, this survival data. And then you could also try to see how the cells, which of the combinations, how in relation to the survival. And here you could see that this was on HeLa cells, this cell line, and you could see that most of the combinations that were predicted by the algorithm all have the trail and the GSK combination. So you could see that the nested structure is embedded even in the predictions if you look at the data over time. And then your question on, I think, on the predictions of why we could not, yes, so this is a good example of what I'm saying. If we were to test how good the model is with a neural network, so this is just doing like an autoencoder, and this is doing a t-SNE with a neural network and a PC with a neural network, you see that in terms of recapturing the initial space and number of original data, it's more sensitive. So even though the autoencoder is very useful and saves a lot of time, it doesn't necessarily mean that you are capturing it's the most sensitive.
Right. Thank you, everybody, for your interest and lively questions. Thank you to all the speakers. And see you after coffee, if you can face any more of me.